our next flash presentation will be Nemanja Koljančić. Okay, sorry for that. That will be speaking about food metabolomics by GC GC-TOF MS and Tandem Ionisation, understanding the impact of climate event on edible corpse quality. Please.
Thank you. Hello everyone. Today I'll speak about our study that we are currently doing during my stay at the university in Turin under the supervision of Professor Cordeiro. And it's about the food metabolomics by GC GC-TOF MS and Tandem Ionisation. So what was the goal of our study? We wanted to understand the different climate effects on the detectable metabolome in hazelnuts, peanuts, and soy crops, and to understand a better correlation between the crops quality, crops breeding, and the different climate events. So we choose these samples simply because they are in everyday diet, and since they are rich in vitamins, fibers, proteins, sugars, and healthy fats. On the other hand, there is a prediction that by the 2050, the world population will increase to more than 11 billion, and thus the crop production will need to increase to more than 50% in the next few decades. So for this approach, we used untargeted and targeted fingerprinting of metabolites that are included in Maillard reaction. They are the precursors of aroma compounds, and they belong mostly to the group of amino acids and sugars. It was, and it's still a big job in front of us since there is a lack of systematic investigation in this field. So how we started? Since we are dealing with compounds that belong to the different groups, and sometimes they can be also in trace level concentrations, we had to use some reliable and highly efficient sample preparation, of course. And we adopted the method from the nature protocols for biological fluids that was thanks to the expertise of the colleagues at the Max Rubner Institute in Karlsruhe in Germany. In practice, what we did, we actually removed by the extraction lipids from all of our samples. Then we proceeded with extraction of our primary metabolites with simple solvent water methanol, and we exposed these extracts to freeze drying. We were left with some solid stuff in the vials, and then we performed methoxymation and derivatization with BSTFA of these extracted primary metabolites. After that, our samples were ready to inject, and for this method, for the GC-GC analysis, we used a fluid-type thermal modulator. I'm sorry, with a 60-meter DBF5 non-polar column in the first dimension, in order to achieve high efficient separation in the first dimension, and mid-polar 1.3 meter column in the second dimension connected to the Tandem Ionisation TOF MS. So basically we had parallel ionization with 12 and 70 electrovolts, 50 hertz per channel. And for our sample, for the first sample, for the first sample set, we had hazelnuts from four regions, two from Turkey, from Akçakoça and Giresun. All of these samples are top quality products. So we had two sample sets from Giresun and Akçakoça, and two samples from Italy, from Lazio and Piedmont. Also, you can notice that we had a fifth batch, which is Giresun. Since in Turkey, traditionally, the post-harvest treatment is performed in field. So they dry the hazelnuts in field in shells during the sunny days. But sometimes some undesirable conditions, such as rainy periods or high humidity, could occur, and it could damage the crop's quality. And that sample set we collected and we had it. So that's actually it. And what we got, these are the chromatograms that we got with enlarged portion region of the chromatogram where the primary metabolites are mostly located. We found 281 peaks, 72 peaks we tentatively identified, and 60 of them were of primary metabolites. And we performed some statistics on these samples, and you can see fine discrimination between the bad post-harvest with the high humidity and the standard post-harvest procedure. Also, you can notice that there is some signs of clusterization on the left side, where are located all the samples with standard post-harvest treatment, like in the case of Lazio samples, which could be the result of the different drying procedures in Italy and Turkey. And we wanted to investigate how these effects actually, how that affects the primary metabolites in bad and good drying procedures, since the variance is given by mainly by the post-harvest treatment. So we compared actually the standard post-harvest treatment and the bad post-harvest treatment from the samples in Giresun, and we could see clear discrimination. And by the box plots, we try to see where is the statistically higher content of the primary metabolites. And the higher sugar concentration were associated with seeds that were actually bad post-harvested. In the same samples, like oxalic acid and similar compounds were in lower concentrations. Next sample set that we had were peanuts. Peanuts were collected. We got the peanut samples from 2 countries, from United States and from Argentina. And you can see there is labels STD and split. That means the seeds were either splitted or they were standard seeds, not splitted. Splitting usually occurs during the germination process when some off flavors could also occur. And the bad and good labels are just the indication how the sample tastes according to the industrial panel test. We got our chromatogram with 530 features. I could identify 152, and 37 were primary metabolites. And these are the 2 regions where most of the primary metabolites from the group of amino acids and sugars were located. So what did we get with these samples? In this case, we could show on the PCA plot that the highest influence on their discrimination was given by their origin, actually. On the right side, you can see the Argentinian samples, and since they are considered to be high quality samples, gold standard samples, because of their peculiar quality, the samples on the left negative plot could be correlated with overall worse quality, but that does not mean that they are bad samples. It's just the clear discrimination between these 2 kinds of samples. USA normal and split are located are clustered together, while the USA bad samples are also separated on the left side. In this case, the box plot showed that the concentration of amino acids and disaccharides was larger in the samples from Argentina, while the USA samples were mainly characterized by monosaccharides such as glucitol and mannitol. The third sample set that we had were soybean samples, but since I think I'm running out of time, I will just briefly tell you what we did with these samples. We basically had two groups of samples. One was infected by Halimorpha halis, or brown marmorated stink bug, which is unfortunately now very popular in the last decade. And another samples were regular soybean samples. And we compared what is the difference in primary metabolites regarding these two kind of samples. I would like to warmly invite you at the poster where we can discuss it in a more detail if someone likes. And in the conclusion, the untargeted targeted fingerprinting proved to be very useful in highlighting the effects of some climate conditions on different crops. And this technique could provide us with data with huge information capacity. I also mentioned that we used both ionization technique, parallel ionization with 12 and 70 electrovolts. But for now, soft ionization we use only for identification purposes. But we are planning to explore the discriminating power also of the soft ionization. Regarding the samples, it showed that suboptimal conditions such as increased water activity or geographic origin or samples damaged by some bug could be correlated with a higher abundance in mono and disaccharides. And also, I would like to highlight that monitoring of the composition and concentration of primary metabolites in crops could for sure be used in agricultural and industries to improve the quality of crops, for example. At the end, I would like to thank Professor Cordero for nice collaboration with her team. Of course, they are doing a really good job, and to present this part of the research, which probably will continue in the next period. And of course, to my professor in Bratislava, and my supervisor, Professor Španik. And also, you can see the logos of our collaborators at the slide. Thank you.